My name's Jeremy Fry. I'm an immunologist and I'm the Director of Sales here at ProImmune, based in Oxford in the UK. You'll have a little bit more of a chance to hear more about ProImmune later on in the talks. But just to introduce us, we're an immunology products and services company that's led the field of characterizing both wanted and unwanted T-cell immune responses for more than 20 years. This has meant that we really have had the privilege of being directly involved in truly groundbreaking immunology. That's from basic science in the mechanisms, for example, of how T-cells function, to the development of the first gene therapies and the latest biotherapeutic drugs, to also playing our part in combating cancer, autoimmunity, and infectious diseases, including COVID-19. And we are honoured to be hosting this expert symposium today on the topic of the immunology of SARS-CoV-2 virus. In the space of just 16 months since the virus was first identified, something's been achieved that many would have said was impossible. Groups from around the world have collaborated to move from the earliest stages of sequencing the virus to the extraordinary position of developing, trialing, and achieving regulatory approval for not one, but numerous efficacious vaccines. Hundreds of thousands, perhaps even millions of lives will have been saved. Even immunology language and expressions that were once just the realm of our research groups have now become household phrases. Now, despite this unprecedented success of the vaccine, communities around the globe are being ravaged by the virus. Vaccine inequality, manufacturing and supply challenges, as well as vaccine hesitancy or even skepticism, are all playing their part in extending suffering and shortening lives. There is a huge amount of work still to do. So our meeting today seeks to educate, to question, and to spark new ideas and connections in the field to leave a lasting legacy. I would first and foremost like to express my thanks to our expert panel of speakers and panelists who have given their time to prepare and share their latest data and thinking today. Just a few points about housekeeping of the meeting. So as you can see, you are all on mute. You will have the ability to unmute. So if we have questions, I just ask that those questions for the speakers be directed to the chat box, and then we'll be able to ask those speakers when they're available to answer those questions. Obviously, there are a lot of people, and so it's going to be impossible to answer all the questions, but we'll do our best to choose a few. The format of the day, you've probably had a chance to look at the program, is I think quite exciting and quite varied. So we have short 20-minute presentations from each of our speakers, and then that will be followed by 5 minutes of questions. So as I say, there'll be a chance to type your questions into the chat box for our speakers. And then at the end of the first session, we will be having a panel discussion, more like a fireside chat, for both the speakers in that session and also bringing in a couple of extra people as well who are experts in the field. And those panel discussions are going to be 30 minutes, and we're going to be putting kind of more of a open questions to our panelists to understand and to get their perspectives on really the pandemic, but particularly the immunology and some of the key questions that remain unresolved and outstanding, which I think will be really exciting part of the program. In the middle, we'll be having a short 15-minute break. We've put that as a networking break in the program. So if you feel that you've already stretched your legs enough and you just want to kind of carry on, there will be the opportunity to network, which is going to be interesting. So we'll put some breakout rooms together and people will have a chance to meet other immunologists from around the world, which I think is also a great opportunity to have a chance to meet other people. So that would just be a 15-minute break in the middle, and then we'll carry on with the second part of our program. So without any further ado, I'd like to introduce our first speaker and make sure, yes, Professor Shin is here. So that's fantastic. So Eui Cheol Shin, our first speaker, received his MD in 1996 and then his PhD in 2001 from Yonsei University in Seoul, Korea, and did his postdoctoral training at the NIH, Bethesda, Maryland. And then he joined the Graduate School of Medical Science and Engineering at KAIST, the Korea Advanced Institute of Science and Technology in Daejeon in 2007, where he is currently a professor. His laboratory, which is the Laboratory of Immunology and Infectious Diseases, focuses on the research of T-cell responses in human viral disease and cancer. It's my great pleasure to welcome Eui Cheol here today, and the title of his talk is Phenotypes and Functions of SARS-CoV-2-specific T cells. Over to you, Cheol, if you want to start sharing your screen. Hopefully you can do that.
Yeah, thank you for kind introduction. Yeah, let me start my talk. So today, I'm going to talk about the phenotype and function of SARS-CoV-2-specific T cell. So in my talk, there are two little bit different topics. The first one is phenotype and function, and the second one is differentiation status of SARS-CoV-2-specific, the CD4 and CD8 T cell. So let me start the first topic. So last year, after the emergence of COVID-19, the many studies have been published. So not only in biology and immunology, but also in any kind of related area. Just 1 second. Yeah. So many studies, even the single-cell RNA-seq analysis was published. So this single-cell RNA-seq analysis included also immune cell, including T cell. So from this data, we can also analyze the phenotype of T cell. So also our group, we analyzed the PBMC from COVID-19 patient by single-cell RNA-seq analysis. Last year, it was like May or June. So now I'm showing you the part of the single-cell RNA-seq data, so CD8 T cell, the part. And now I'm showing you that, so in this case also we included not only COVID-19 patient PBMC, but also severe influenza patient PBMC, which were acquired pre-pandemic the year. And I have to allow the participant to enter this session. Let me, because I have now I'm a host. That's the reason why. Yeah.
No, you don't need to worry, Cheol. It's okay. We can admit people.
Yeah. So in this single-cell RNA-seq analysis, we found that for example, cluster 1 is more enriched in the severe influenza patient. Or for example, the other cluster 3, this one's more enriched in COVID-19 patient. In the subsequent analysis, we found that what is the influenza specific cluster 1 and COVID-19 cluster 3, they have a different molecular pathway, and they have a different gene ontology molecular pathway. But meanwhile, so I realized that this kind of analysis is somehow meaningless, because in this assay, we don't have any information about the antigen specificity of the T cell. So there are some quite difference between the influenza patient the T cell and COVID-19 patient T cell, but we don't know these are bystander cell or COVID-19 SARS-CoV-2 specific T cell or influenza specific T cell. So without the antigen specificity, this information, somehow these information very limited. So meanwhile, the last year, so many groups also reported the function of the SARS-CoV-2 specific T cell from COVID-19 patient or convalescent. For example, the La Jolla Institute, Shane Croce and Alexander Rosetta group, they published it exactly 1 year ago with such data. They examined the COVID-19 convalescent by performing the AIM assay, activation induced marker assay, and they reported that CD4 and CD8 T cell response nicely elicited during the COVID-19 by using the OLP stimulation. So this kind of the detection method of the functional T cell, so we need to stimulate the PBMC or T cell by the OLP or protein in vitro stimulation-based assay. So these assays include not only this AIM assay, but also gamma ELISpot assay or intracellular cytokine staining or like that. But yeah, sure, this technique is very sensitive technique. This technique allow us to detect very easily the SARS-CoV-2 specific T cell. But one of the pitfalls of this assay is that the fact that by using this assay, we cannot examine the phenotype of SARS-CoV-2 specific T cell as it is, because during the in vitro stimulation, the stimulation can change the phenotype of this virus specific T cell. So our group, we wanted to examine the phenotype of SARS-CoV-2 specific T cell in patients, so without changing their phenotype. The only way was the MH class 1 tetramer, or class 1 or class 2. But my group, we are much more interested in the CD8 T cell response. So we decided to use MH class 1 multimer. It was last year, like July. In that time, we recruited more than 100 patients, Korean patients, SARS-CoV-2 patient and convalescent, and we acquired the PBMC. So, oh yeah, we acquired 200, and around among 200, the more easily we can apply this MH class of multimer to HLA-A02, the patient. So among 200 patients, 100 patient was HLA-A02. So we could analyze this, the 100 patient by using MH class 1 multimer. And in this assay, in that time, we didn't know which epitope is more immunodominant epitope or not. So we just blindly, we obtained all the commercially available, the multimer in that time. It was like June or July last year. When we just blindly applied these 8 different the multimer to patient sample. And by using this the multimer, we detect the phenotype with the multicolor flow cytometry analysis. But at the same time, we could combine the multimer staining with the functional assay, cytokine secretion assay. The cytokine secretion assay is very similar to intracellular cytokine staining. So we could combine this image class of multimer and the CSA assay. And now I'm showing you one of the representative data. This one is showing the data from S269, this S269, this is the multimer. And also this one is the immunodominant one because among 112 patients, around 40 patients has a positivity. So one of the dominant one. At the second one, we found that the S1220, this one is more the minor epitope. And anyway, among these 8, in the blind application, we could find that these two epitope is the working. So in this study, we used the 2 multimer. At the same time, we found that this S269 and S12, the 1220, these 2 epitopes have the limited homology with the common cold coronavirus. So we also tested that in unexposed individual, we could not detect any of these tetramer positive cell, the multimer positive cell. After successful detection of this SARS-CoV-2-specific CD8 T cell, we examined the phenotype of this SARS-CoV-2-specific CD8 T cell. And so we found that in early convalescence phase, almost all of them were the effector memory cell phenotype, and some of the considerable proportion of these SARS-CoV-2-specific T cell, PD-1 positive, and also also at the same time, they exhibit activated phenotype with the CD38 positive and HLA-DR positive. And in terms of frequency, severe patient has much higher, significantly higher frequency of this multimer positive cell. But as of in this study, we want to answer this very important question. The last year, after the emergence of COVID-19, the many studies they reported that SARS-CoV-2 specific CD8 T cells are functionally exhausted. So these 3 papers were published like last year, March or April. But they just reported the PD-1 expression among CD8 T cells. And then just conclude that maybe due to PD-1 expression, the regulation of PD-1, maybe they are functionally exhausted. So also in our study, in acute stage, we found that, especially the data was obtained from the acute stage, and almost all of the SARS-CoV-2 specific CD8 T cell, the multimer positive cell, nearly 100% are PD-1 positive. So really, the SARS-CoV-2 specific T cell were PD-1 positive. But can you say that due to the PD-1 positivity, all of them are functionally exhausted or not? That was our question in that time. So we combined, to answer this question, we combined the multimer staining, the MH class and multimer staining with the CSA, the cytokine secretion assay. With this analysis, we could analyze the PD-1 negative cell and positive cell, which cell population produce interferon gamma. In this way, we analyzed. And we found that even the interferon gamma positivity was much higher in PD-1 positive cell population compared to PD-1 negative cell population. So to make sure, even we sorted first the multimer positive cell from PBMC, then we further sorted PD-1 negative population and PD-1 positive population. Then we stimulate them with the peptide, and we perform CSA, cytokine secretion assay. So we found that interferon gamma was nicely produced irrespective of, regardless of PD-1 expression. We confirmed such data again by using very hyperacute stage of the patient sample. So PD-1 positive cell nicely produced interferon gamma. At the same time, they exhibited the CD38 HDR very activated phenotype. So with this data, we could conclude that SARS-CoV-2-specific PD-1 positive CD8 positive T cell are not exhausted but functional. So we conclude that this not only exhausted T cell but also activated T cell can upregulate PD-1. So that's the reason why this SARS-CoV-2-specific CD8 T cell express PD-1. And they at the same time, they can produce very nicely interferon gamma. And in 7 patients, we have some longitudinal serial sample acquired from acute stage. So we could perform kinetical analysis of this SARS-CoV-2-specific CD8 T cell by using this multimer. And this the gray area is representing viral titer. And if the viral titer decreased, the multimer, the frequency of the viral specific T cell, accordingly, it also decreased. So it followed the viral titer. But at the same time, KAIST, the proliferation marker, or 38DR activation marker, it is accordingly decreased. But the cytotoxic molecule like purpurine enzyme is more sustained. In that way, we could analyze the kinetical change of SARS-CoV-2 CD8 T cells phenotype. So let me summarize this in my first part. In my first part, so we apply this MH class 1 multimer to detect SARS-CoV-2 CD8 T cell without any changing the phenotype, without any in vitro stimulation. Sure, in the functional assay, we stimulate them in the CSA, but at least for the phenotype analysis, we didn't stimulate the cell to PBMC. So as a result, we conclude that these SARS-CoV-2 CD8 T cell, they upregulate the activation marker, proliferation marker, and cytotoxic molecule. At the same time, these CD8 T cell upregulate PD-1. But however, they produce interferon gamma very well, so functionally not exhausted. So that was our simple conclusion of my first part. In my second part, I'm going to show you the differentiation status of SARS-CoV-2 CD4 and CD8 T cell during 8 months follow-up. So in this study, we examined the follow-up sample for 8 months after recovery from the COVID-19. So first, we perform ELISpot assay by stimulating PBMC with the OLP covering spike, membrane, or nucleocapsid. And overall, we found that the T cell response is well sustained for 8 months, more than 240 days after the infection. And in the statistical analysis, so we divided these 8 months into the 3 stages, 3 phases, and there's no decrease in statistically. So we conclude that the SARS-CoV-2 T cell response is well sustained for 8 months. And now I'm showing you some patient data whose sample is longitudinally acquired. So up to the 250 days, there's not so much decrease of this T cell response. We confirm this data by AIM assay, activation-induced marker assay. So in activation-induced marker assay, to detect antigen-specific CD4 T cell, we use OX40 and CD137. For CD8 T cell, we use CD69 and the 4-1BB. So in this AIM assay, we confirm that both CD4 and CD8 T cell response is relatively well sustained for 8 months against S or M or N, irrespective of a viral antigen. But in the AIM assay, we decide to analyze the differentiation status of SARS-CoV-2 T cell by staining them with the CCR7 or CD45RA. As you know very well, with these 2 markers, we can determine their different status, such as central memory or effector memory or TEMNA, like that. So now I'm showing you the proportion of the central memory, effector memory, and TEMNA and naive-like cell among the SARS-CoV-2 CD4 T cell. Mainly, the central memory cell are one of the predominant population, and TEMNA, the most terminally different cell population, was scarcely detected. But meanwhile, we found that considerable proportion of the CD4 T cell, virus-specific CD4 T cell, has naive-like phenotype, like CCR7 positive and CD45RA positive. So this proportion cannot be ignored. So the grad student, he asked this question: what's the nature of this naive-like population? So he hypothesized that maybe this naive-like population is the stem cell-like memory cell population, because the Tscm, the stem cell-like memory cell, they have the naive-like phenotype in terms of CCR7 positive and CD45RA positive. But one of the difference between real naive cell and Tsm is the CD95 positivity. So we applied also this the CD95 staining, and we found that almost all the 100% of this the naive-like population is Tscm. So now I'm showing you the proportion of Tscm among SARS-CoV-2-specific CD4 T cell, and now I'm showing the change during the 8 months. As you can see here, the SOM or N-specific CD4 T cell or CD8 T cell, the considerable proportion of the antigen-specific T cell, memory cell, stem cell-like memory cell. So we confirmed that this the Tscm phenotype by using the MH class of multimer. We detected this the S269 multimer positive cell, and we combined this the multimer staining with the CCR7 and CD45RA and CD95. We found that around the 40% of this the T cell are Tscm in the SARS-CoV-2 multimer positive CD8 T cell. But in the healthy donor, flu or CMV space among these T cell, nearly 0% of them are Tscm. And also we found that among Tscm, there are 2 different populations. It's known that the PD-1 and according to PD-1 and TIGIT positivity. So PD-1 and TIGIT negative population is known as more stem-like one, and double positive one is known as a more exhausted-like one. And we found that SARS-CoV-2 specific CD8 T cell, the Tscm, they are much more the PD-1 and TIGIT double negative cell population. It means that they are more strong stem cell-like feature. And also we confirmed that really the SARS-CoV-2 specific Tscm cell have real stem cell-like capacity. In vitro experiment, we found that so they have more antigen-driven proliferation capacity. Also, they have multipotency. It means that after the in vitro stimulation with the antigen, they can differentiate into the diverse population. Also, upon IL-15 stimulation, they have a high self-renewal capacity. So the SARS-CoV-2 specific Tscm cell, they have all 3 aspects of Tscm in terms of proliferation and multipotency and self-renewal capacity. And as a result, we found that even at the long term after, like even day 221 since the viral infection, CD8 T cell or CD4 T cell, they can maintain their polyfunctionality and proliferation capacity. So they are very polyfunctional. And in the CTV, the proliferation assay, they show the very vigorous proliferation capacity upon in vitro stimulation. So let me summarize my second part. In my second part, we examined the long-term longevity of this SARS-CoV-2 specific T cell response by the diverse T cell technique. And meanwhile, we found that the Tscm, the stem cell-like memory T cell, are well developed, and they have the typical function of the Tscm, the high proliferation capacity and self-renewal capacity and multipotency. So far I showed you the topic, the phenotypic function and differentiation status. Then before closing my talk, let me just briefly mention about the importance of the T cell study. So currently, the variant of concern, there are many variants of concern, is now emerged. So now it's also known that the vaccine-induced neutralizing antibody, this neutralizing activity is attenuated against the variant of concern. But still, the T cell, we have T cell, vaccine-induced T cell. So T cell can target multiple epitopes in the viral antigen. So we need to study more and more about the vaccine-induced SARS-CoV-2 T cell response. Also at the same time, so Antonio group and also Shane Crotty group, so many groups also reported that even without any exposure to SARS-CoV-2 among unexposed individuals, there's a considerable proportion of them have SARS-CoV-2-reactive T cell response. Then what's the clinical meaning of the SARS-CoV-2-reactive T cell response? We don't know yet exactly. So we need to know this answer. in near future. So in that way, we can overcome this pandemic. So this work was done by Dr. Ryan, the Min-Seok Ra in my lab, and also Jung-Seok Lee and Jae-Young Jung, Arum Kim, and Jun-Young Go, and Garam Nim, with the collaboration with the Korean, the clinician for the clinical sample. And thank you for your attention.
Thank you very much indeed, Eui Cheol. That's fantastic. Really appreciate that. I'd like to open the floor for questions. If you'd like to type your questions into the chat box, please do that. While we're waiting for those to pop up, Eui Cheol, just a quick question then in relation to the sort of stem cell-like capacity of the SARS-CoV-2 specific CD8s. That's something which is obviously, I don't know whether you've extensively investigated that in other viral diseases, but obviously your experience with HBV and other viral diseases is extensive. So why do you think that the SARS-CoV-2 is kind of different in regards to different viruses in the stem cell-like T cells?
Yeah. So in human viral immunologic study, the Tscm was the more extensively studied in previously in yellow fever vaccination situation. In the case of yellow fever vaccination, the Tscm is well developed, like SARS-CoV-2 infection. So the Tscm, the development, it means that indicate that the memory T cell response maybe will be maintained for long term in future. So that was the proven in yellow fever vaccination. But in my data, I showed you that for example, CMV-specific T cell, there's not so many Tscm. So maybe this is due to immunosenescence of CMV-specific T cell. Because in the case of CMV infected people, the latently infected individual, CMV-specific T cell is repeatedly stimulated by the chronic stimulation. So due to this, the repeated stimulation, Tscm is the depleted. I can say that. Yeah, like depletion. Yeah, I can say that. Yeah.
So do you think that in the context of additional exposure to different variants of SARS-CoV-2, that actually might reduce the Tscm frequency?
Yeah, too many times, then maybe if too many times, then it'll be reduced. But just 1 or 2 or second, this kind of exposure may not. Yeah.
Thank you. That's great. Any other questions? People are getting warmed up still.
Can I ask directly a question? Because I was
Please do. Who's speaking?
Antonio Bertoletti.
Antonio,
sure.
Nice to have you with us. Would you like to ask your question?
Yeah, no, I mean, the first question actually was exactly what you asked, Jeremy. But the second one is, I was noting in your frequency of CD4 and CD8, it looks to me that the CD4 are really sort of increasing of frequency during time, whereas the opposite is with CD8. Can you confirm this sort of behavior that the CD8 are really sort of, let's say, going down as a frequency, but the CD4 seems actually to increase? Because these are all, has been also seen by others. So I'm just interested to.
Yeah, that's a very good question. So at first, I have to say that point out that I have the much more previously more experience as an acute viral infection. I have many experience with the HCV infection, hepatitis C virus infection, but compared to HCV, SARS-CoV-2, much more CD4 predominant. It means that in HCV, there are much more CD8 T cells, but in SARS-CoV-2, I don't know the reason why, but CD4 T cell is much more as an antigen-specific T cell. But and not only immediately after recovery, but also like now Antonio's mentioned, during the follow-up, CD4 T cell is well maintained relatively, but CD8 T cell is relatively not so well maintained. So there's more somehow CD4 dominancy in SARS-CoV-2 infection, but I don't know the reason why. Yeah, you are right, Antonio.
Yeah. No. Thank you.
Thank you very much, Antonio, for your question. And Eui Cheol, thank you very much indeed for your presentation. Please do stick around because we've got the panel discussion later on, so you'll be able to ask more questions there. So thank you very much indeed.